let's make a start. I just want to welcome everybody for being here tonight. We're really excited that ISCAST and New Zealand Christians in Science have teamed up again to bring you 10 fantastic talks on topics that intersect science and Christian belief. I'm Sarah Wilson. I'm the Program Director for ISCAST, and I've been working with Chris Mulheron, who is the Executive Director of ISCAST, and Nicola Hoggard-Cregan, the Co-Director of New Zealand Christians in Science, to bring you these weekly conversations. Before I introduce our speaker tonight, I'd like to invite Nicola to open in prayer for us, please.
Thank you, Sarah. Yes, and a very big welcome from New Zealand Christians in Science as well. We're really glad that you're all here and that we can journey with you into December. So let us pray. Loving and Creator God, we thank you for the abundance and absolute wonder of this world, and for the many avenues of scientific investigation and exploration we have represented here and in the wider community. We thank you for that this wonder gives us some comfort in these straitened times. And we thank you also for the opportunity to reflect on all of your creation and its multiple forms, and especially tonight on some of the smallest and most numerous creatures in the species of insects, and on the ways of entertaining and glimpsing your love as through a glass darkly. We ask your Spirit to guide us in our thinking and in our listening, and we lift up Professor Batterham to you as he presents to us tonight. In the name of Father, Son, and Holy Spirit. Amen.
Amen.
Amen. Thank you, Nicola. Yes, we have got Professor Phil Batterham tonight, which is wonderful. I just wanted to let you know a little bit about his background. Phil earned his bachelor's degree with honours at La Trobe University and then completed a PhD in genetics at Monash University. He did a postdoc in the US for a couple of years and then came to the University of Melbourne as a research fellow before being appointed as lecturer in genetics. Over the years, he rose to the level of professor and has been recognised at the national level for his research, teaching and science communication. The Genetic Society of Australasia awarded him the M.J.D. White Medal for Research Excellence in Genetics. Phil was also awarded the Carrick Citation for his exceptional record in supervision, support, and career mentoring of research higher degree students. And he has been a 5-time finalist in the Eureka Prize for Public Communication of Science. Phil has organised several major international conferences, including the International Congress of Genetics and a UN conference on global health. He is the current president of the International Genetics Federation and a fellow of ISCAST. And Phil will be talking to us tonight on insect Armageddon. So I'll hand the floor over to you, Phil.
Thank you. So it's a great pleasure to be here. And I'm just going to try and get the format of this right for me to see what I'm talking about. So it is a great pleasure to be here. I'm honoured to be asked, and added to the introduction, I became a Christian. I was from a non-Christian family, but became a Christian when I was very young, and so I've lived a life of science and being a Christian in parallel for a very long time. So, problem number one, my slides are not advancing. So, I live on the northern fringe of Melbourne in a suburb called South Morang, and it's the traditional lands of the Wurundjeri people, and I pay my respects to their elders past and present and to any Indigenous people that might be here in the audience. As a young Christian, I began my research career in 1976, and I was always asked this question: given what I was researching, which was in the area of population genetics, well, what about Genesis? And I would be quite intimidated because in those days there was a deep division, it seemed, between church and science. And I think it would have been very difficult for me to give a talk of the nature that I'm giving tonight to any Christians at that point in time. But I've become very much more interested in the harmony of my faith and science. And so I'm tonight presenting something of my journey, where I'm at this point in time. So I believe that Genesis provides insights into who our God is, and that God is both the creator and he's the God of truth. I think science is noble because it's a search for truth. And I believe that evolution is the mechanism by which God created. However, if there are those present that don't believe that, then whether you believe that creation occurred in a period of billions of years or in 6 days, so much of what I'll say tonight applies. And the final point I think is really important, that God has given us responsibilities to care for his creation. Now, diving a little deeper into Genesis, it starts with these words: "In the beginning God created the heavens and the earth. Now the earth was formless and empty, darkness was over the surface of the deep, and the Spirit of God was hovering over the waters. And God said, 'Let there be light,' and there was light." Those verses and the ones that follow really talk about how God created order from chaos. But so much of human history, recent history in particular, has shown humans creating chaos from order. We often talk about God creating plants and animals and other organisms, but God in fact created ecosystems. And in ecosystems, all organisms are interdependent. And they have a capacity to adapt to environmental change, albeit slowly. And when the environment changes rapidly, organisms can be caught short, unable to adapt to those changes. Humans have had an impact across all ecosystems, and yet our survival depends upon ecosystem health, and we tend to convert ecosystems that look something like the picture on the left to something that looks more like the picture on the right. My next point is that my genetics tells me that all forms of life are really related. So some organisms have DNA genomes, some have RNA genomes, but there is a relationship. And even organisms that seem very different from us can have a fairly close relationship. And because I'm going to talk about insects tonight, I want to just highlight this organism, which the Americans call the fruit fly, we call the vinegar fly. And it has a very high percentage of its genes in common with humans. And because of that genetic similarity, there have been, in fact, 6 Nobel Prizes in medicine awarded for research conducted exclusively in this fly, Drosophila. And that's research that's been relevant to human development, to behavior, to all sorts of other traits. And there seems to be no end to the utility of this organism, and I'm expecting that more Nobel Prizes will be awarded. So I see there being a kinship in creation. When I reflect on Genesis, I think that God is actually the original greenie. And there are verses there that we often we tend to overlook. So in the creation story, God says that plants will be our food. They'll be our food and the food for all of the beasts of the field. It's interesting when you read the Bible to, in fact, contemplate what is not said. And there are a couple of things that are absolutely not said in Genesis 1. Firstly, you don't find any permission to kill or to eat animals. You don't find any permission to cut down trees. And so with that as a background, I come to insects. And this image that's before you shows the extraordinary diversity that there is among the insects of the world. Current estimates are that there are 5.5 million insect species, and the vast majority of these are not pests, and this becomes relevant later. Unfortunately, there is now evidence that population sizes of land insects is shrinking by an average of 10% per decade, and that trend has in fact persisted over several decades. So there's been a precipitous decline in the size of insect populations. And this has been picked up both in journals like Science and in newspapers like the New York Times and many other publications. There's much emphasis on honeybees, and I'll come to that a little later. National Geographic asks a question or makes a statement, you'll miss them when they're gone. I often have to explain to people why insects might be important, and I'm sure that many in this audience would know why, but there's a number of reasons. Firstly, there are insects like honeybees that are pollinators. There are other insects that control pests, such as parasitoids. Insects are very important in nutrient recycling. They are a food resource in ecosystems, which we'll look at a little more in a moment. And in fact, they're becoming a food resource for humans, and there are many other layers to the benefits that insects bring. Insects, which are part of the larger family of arthropods, make up a huge percentage of the biomass of this earth, dwarfing the contribution to biomass of humans. But when we get to a more important level, looking at this energy pyramid here on the left, you can see that at the bottom layer of the pyramid, there in fact are the producers of energy, which are plants. And in the layers above the plants, you see the consumers of energy. And at the second bottom layer, the primary consumers are in fact insects, which are food for the secondary consumers, often small mammals, which are food for the tertiary consumers, which are birds and organisms like us. Now, in this pyramid, if you destroy the bottom layers, then you have a similar effect to pulling out too many blocks in a Jenga stack. And so when we're talking about the loss of insects, then we can expect that we'll see impacts on organisms that are higher up the pyramid. And so it's been noticed in the United States, where they've got accurate data, that 2.9 billion birds have gone missing since 1970, or more precisely, population size of birds has decreased by 2.9 billion, or by 30%, and in many cases that has been directly linked to insect decline. Now, the reasons for insect decline are many. So we can see on this diagram, which comes from proceedings of the National Academy of Science, you've got causes like climate change, which is the obvious, but there's drought, there's nitrification of soils, there's pollution, there's introduced species, there's land clearing, there's the intensification of agriculture, deforestation, and insecticides down here. Now, I'm going to talk a little bit about climate change and a lot more about insecticides. Recently, I heard someone in comment about climate change as being the loudest canary in the mine. And the reference to canaries in mines is miners of old used to take canaries down in the mine, and if there were noxious gases being emitted, then the canaries would die before the humans would know to get out. So in saying that the climate change is the loudest canary in the mine, we're actually saying that it's one of many factors, many aspects of environmental decline, but it's the one that's getting the most attention. The reference to 1 plus 1 equals 11 is a reminder for me to tell you that these factors don't act in isolation and that they may synergize and have multiplicative effects. So let's talk a little bit about climate and the impact it will have on insects. Insects don't control their own body temperature. They're cold-blooded, so their internal temperature is determined by ambient temperature. All biological functions, from metabolism to movement to behavior to reproduction, are regulated by temperature. And a given insect species has a precise range of temperatures over which it can survive and reproduce. I'd note that in many insects it's been shown that males become infertile at temperatures several degrees below that which an insect species can survive. And so at quite modest temperatures, populations may crash simply because of male infertility. So as a consequence of all of that, climate change can lead to range shifts. It can lead to some species expanding their range and others contracting until they have no range at all. So turning to insecticides, a really important thing to remember is that there are no insect pest-specific insecticides. And in fact, insecticides are designed to target receptors in insects, often in the nervous system, that are found in all insects. And the second point on this slide is that massive amounts of insecticide are used globally every year, and these insecticides have impacts at minute doses. So before we talk about the downside of insecticides, we need to talk about why they're important, because one might simply reach the conclusion, well, we can just stop using insecticides immediately. Well, the reason we need insecticides is because we need to feed the world. And the global population is increasing by 220,000 people per day, but the land available to produce food from is decreasing by 33,000 hectares per day. It's a very ugly ratio that's continually changing in a bad way. And insecticides increase crop yield, depending on the crop, from 6% to 79%. And in Australia, on average, it's increasing yields by about 29% and brings tremendous economic benefit to our nation. But on the downside, low-dose exposures can have profound effects. And insecticides have been are being blamed as a cause of so-called colony collapse disorder in bees. And they probably are a contributor, although there are other contributors. The number one enemy of the bee community in terms of insecticides is a neonicotinoid insecticide called Imidacloprid that I'm going to talk to you about. It's been banned from agricultural use in the EU. And when I get to the particulars of the research, I want to talk about work published in the Proceedings of the National Academy of Science last year, with the lead author being my student shown here, Felipe Martelli. I'm going to tell you about two insecticides, one which initially appears as the Darth Vader insecticide, the bad guy, that's Imidacloprid that I already mentioned, and one that appears to be the good guy, Luke Skywalker, and that's Spinosad, which is an organic insecticide. So, as already mentioned, many insecticides target the nervous system, and Imidacloprid and Spinosad certainly do. They target these nicotinic acetylcholine receptors. Now, sorry, I was just reaching for a pointer here. Okay, so these receptors actually also form a channel, and cations such as calcium flow through the signal, through that channel, carrying a signal. There is a natural agonist called acetylcholine that keeps the channel open while it binds, but acetylcholine is metabolized by an enzyme, broken down, and the channel then closes. So these channels open for milliseconds and then close, and that's very important. What these insecticides do is they bind at the site where acetylcholine would normally bind, but they are not metabolized, and therefore they bind and the channel remains open, and so the neuron, the cell, is flooded with acetylcholine. Now, low doses of insecticide will change behavior. That's been demonstrated. High doses will cause death. So we don't really know much about what happens downstream of this interaction of the insecticide with the receptor under low dose conditions, and that's what Felipe's work was directed at doing, at finding out. Okay, so introducing that fly again, the Vinegar Fly Drosophila. It's not a pest, it's found everywhere, it's exposed to insecticides, and it's genetically very similar to insects, including insect pests. So Felipe developed a low-dose protocol to examine the impacts of imidacloprid on Drosophila larvae. And his index was to look at the level of movement shown here on the y-axis of the graph over time shown on the x-axis of the graph. He used a dosage of 2.5 parts per million. Now this is way below the 10,000 parts per million used in garden sprays, way below the levels seen in environmental contamination. And he showed that over time the level of movement in these Drosophila larvae exposed to this dose decreased compared to untreated larvae. However, if you recovered these larvae and washed them off and allowed them to develop to adulthood, there was no really significant change in the level of survival, although they did develop a little more slowly. So it was known that if you increase the flow of calcium levels in neurons, it leads to the activation of an enzyme called nitrous oxide synthase, which leads to the production of nitrous oxide, which is a source of reactive oxygen species. Reactive molecules that do what we would call oxidative damage to macromolecules, including fats or lipids, to proteins, and to DNA. These impacts are in the mitochondria, which are energy centers in every cell. So every cell contains mitochondria, and they produce energy that allows that cell and indeed the organism to function. So in looking at mitochondrial function, Felipe had two assays. One was to look at the activity of an enzyme, aconitase, that's in the energy production pathway, and levels of that enzyme dropped precipitously in larvae that have been exposed to imidacloprid for 2 hours. And he looked at ATP, which is the molecule that carries energy, and the levels of ATP also dropped precipitously in just 2 hours of exposure. He could look at ROS quite directly, these reactive oxygen species, and if we focus on this panel here, we see that after 2-hour exposures in the brain and the midgut of the insect, we see a much brighter red stain, and this red stain is picking up levels of reactive oxygen species. turning to a tissue which is the equivalent of the liver, he was able to look at a defense system against the impacts of ROS. In these liver cells, the fly produces these lipid droplets, and basically these lipid droplets harbour lipids so that they will not be impacted by the ROS that might be in the cytoplasm of these cells. And Philippe was able to show that in this tissue, and we'll only look at this tissue, that compared to controls that were not exposed to imidacloprid, that you saw massive expansions in the number of these protective lipid droplets. Looking at gene expression, he was able to show that there were perturbations of metabolism, there was evidence of neurodegeneration, and there was impaired immune function. So we then turned to adults to look at chronic low-dose exposures over multiple days, and he tested a range of doses and ultimately did experiments with this dose of 4 parts per million, where over a period of about 20 days you'd get minimal death, but after that there would be significant death. And he conducted his experiments over this 20-day period where the flies weren't dying in significant numbers, but he would look there for significant biological impacts. And he performed this assay of function called the Climbing Assay. Basically, you have flies in a vial, you bang the vial, and the flies go to the bottom of the vial, and then their natural tendency is to climb. And so you can see here that in black bars, we're seeing flies that are exposed to imidacloprid, and in the white bars, which are non-existent here and here, but here you can see it, a white bar, the climbing performance of flies not exposed to imidacloprid. Now, he looked at flies after 1 day of exposure, 10 days of exposure, and 20 days of exposure, and what we're charting here is the percentage of flies that failed to climb, were unable to climb, and you can see that over time that percentage is increasing dramatically for the flies that are exposed to imidacloprid. There's a very slight increase, but only at the 20-day period, with flies that were not exposed to imidacloprid. Now, this climbing assay is not just an assay of locomotion, because climbing requires coordination, and often a climbing assay in flies is used as a proxy assay for neurodegeneration that we'll come to in a moment. Now, he was able to show that if he treated flies with an antioxidant that we call AD4, that the survival curve changed quite dramatically. So here is the survival curve that I showed you before, with flies exposed to 4 parts per million. The survival curve for the control shown up here in pink, and the survival curve of those exposed to both the imidacloprid and the antioxidant. And this suggested that some of the phenotypes that we're looking at are attributable to the oxidation, the ROS molecules that are being produced as a result of exposure to imidacloprid. Now, since we had evidence of neurodegeneration, we began to look at the brain, and we focused on the eye. Now, here he conducted electrophysiology and produced electroretinograms. Basically, you put an electrode into the eye of the fly and another electrode into the back of the head of the fly, and you can look what happens when you get a normal a light impulse. So you direct a light impulse through this microscope. The fly is on the stage down here, and then there is this characteristic curve, which I won't go into in any detail, but you can see that the curve is very different in a number of aspects between the imidacloprid treated flies and the controls. We're examining at the 20-day mark, and you can see that this so-called on transient is shrunken, and this so-called amplitude, the depth of this curve, is reduced. Putting those together, experts can judge that these flies are in fact blind. Now, we had a closer look at the eye, and before I show you some electron microscope images, this is a surface view of the eye looking down a light microscope. Here, each one of these is a single eye, one of the several hundred eyes making up the compound eye of a fly. And here we're looking at a diagram of what we're seeing here, and basically you've got these photoreceptor neurons labeled R1 to 8, and these dots here, the red, correspond to the red dots, which are the light trapping organs of those cells called rhabdomeres. Now, when we look at flies exposed for 20 days, under the eye is sectioned and we look under the electron microscope, we can see a number of differences between the images that we get for control flies versus the imidacloprid flies. Most stunningly obvious are these areas highlighted in blue. These are support cells that surround these photoreceptor neurons, and these support cells are in fact dying. We see an expansion in the number of mitochondria highlighted by yellow arrows, but the mitochondria upon closer inspection look to be defective. And so with imidacloprid, we've got this picture of failure at multiple levels when insects are exposed to very low doses of imidacloprid. So let's briefly turn to Spinosad, which is the good guy, supposedly, the Luke Skywalker in my story. So this is an organic insecticide. It's deemed organic because it's produced by soil bacteria. It was discovered in the field once in a rum distillery in the West Indies, and it's considered therefore to be organic, even though it's produced in large vats in fermenters, and it's clearly used in organic farming, and it's considered to be harmless to beneficial insects. I'm just going to show you one image of what this insecticide does to the Drosophila brain. I should say that all of the other effects that we saw with imidacloprid, we see with Spinosad. Now, if you look at a control brain, the brain has been sectioned. So we're seeing here the outer rims of the eyes. We're seeing the optic lobes of the brain. This is a control brain at 20 days, and it looks quite normal. When we do sections of eyes of flies that have been exposed to imidacloprid, the brain looks quite normal at this level too. But here's what the brain looks like in a fly that's been exposed to Spinosad at a dose of just 0.2 parts per million over 20 days. 17% of the central brain on average is absolutely blown away, is missing. Technically, we say it's vacuolated. What's happened here is that there has been massive cell death, particularly in these regions of the brain, which we know are important for really important behaviors such as learning, memory, the capacity to smell, and so many others. So imidacloprid is a devastating drug at low dose. And here's an image of the brain. I described the so-called mushroom bodies, and they are highlighted by fluorescence here in this beautiful image of the brain. And so we can see that that region is basically gone. So as I come towards the end of my talk, both imidacloprid, the synthetic insecticide, and Spinosad, the organic insecticide, can be called Darth Vader. Both are devastating in their impacts at low doses on insects. Now, the implication for other insecticides is this. that other insecticides, many of them, have been shown to be generators of reactive oxygen species. And therefore, I believe that if they were studied in the way in which we've studied these two insecticides, we would find that they would have similar impacts at low doses on not only Drosophila, but on beneficial insects that we really value in our environment. So what would these impacts have? What impacts, would these, let's just turn that around. The impacts that we've seen generated by these insecticides, what would be the result of that in the field? Well, energy loss would be really important in terms of high energy demand processes. And they include flight and reproduction, but also the function of the brain itself is very demanding of energy. We've seen a shortening of lifespan, and in some insects that would not be very important, but in honeybees it would be vital because bees, particularly worker bees, change their role in the hive as they age, and so it's important to have a diverse age structure in the hive. Neurodegeneration would impact a range of important behaviours. Loss of vision would impact the capacity to locate food. Lack of immunity would mean they're susceptible to infection. And the changes in gene expression that I've not talked about in detail would reduce resilience in the face of other stresses such as climate change. Now, the doses at which these impacts that I've described will kick in will vary between species, but I believe they'll be found in any species where they are investigated. So further research demands that other insecticides be tested, that we look at impacts on some of these other factors like reproduction and immunity, and that we investigate the possible synergistic interactions between factors such as global warming and insecticide exposure. There are alternate insect control strategies that could be very useful. I won't go through them in detail now, but we might pick up on them in question time. One of them that is very promising is the use of endosymbiotic bacteria, and there's a lab in the institute that I work in, Ari Hoffman's lab, that is a world leader in the use of endosymbiotic bacteria for the control of insect pests. So with that, I'll just leave you with my acknowledgements and then ask for your questions.
Thank you so much, Phil. That's been a wonderful talk. I'm just going to introduce Chris Mulheron, who will facilitate the Q&A. Thanks, Chris.
Thanks very much, Sarah. I'm going to invite William, if you're there, William. I think, William, you had a question, didn't you? So I wonder, would you like to unmute yourself and ask your question? Go ahead.
Hi, William.
Hi, my question is, what does insecticides have in food growing in New Zealand?
What impact will they have on food? Well, William, the insecticides that are used on food are there to stop insects from destroying the food. What we've been talking about is what they might do to other insects that might be in the area when the crop is sprayed.
Thanks, Phil. I wonder if Neil Ormerod is there and if he would like to unmute himself and ask his question.
Yeah, thanks, Chris. Look, acetylcholine's also prominent in nervous systems in animals as well. And, you know, a lot of nerve agents were derived basically from insecticides, the sort of nerve gases that were used in World War II. and are still being used. So the question is, these insecticides, do they impact on the food chain? Do they bioaccumulate? And is there the possibility then of these same chemicals impacting on humans in the long run and producing the same sort of effects even at very low levels?
Neil, it's a good question. I'm not an expert on the bioaccumulation. Imidacloprid, obviously each of these chemicals has a half-life, and so they do degrade. But the question about humans is one that I get asked a lot. As you rightly point out, these receptors are found in all animals, and humans have counterparts to these receptors. The insecticides we know bind to human receptors with low affinity. And so in the short term, there's unlikely to be much impact. However, one of the things that I think about when I consider our research is that many chemicals may be ROS generators. So in our lifetime, we're consuming many different chemicals, and if the bottom line effect of many of these chemicals is to increase levels of ROS, then there could be a cumulative effect. And I should say that what we see in terms of the impact of imidacloprid really does resemble some forms of neurodegeneration that we see in humans. So in some inherited forms of neurodegeneration in humans, the real issue is with mitochondrial function, a buildup of ROS, and neurodegeneration follows. There's another form of neurodegeneration in humans, which is due to so-called lysosomal disorder. So it's due to defects in another organelle that resides within cells, a very important organelle. And it turns out that while Spinosad has very similar effects to imidacloprid, downstream effects, that at its source, it's creating defects in lysosomal function. And so we think that these two insecticides are in the fly phenocopying types of neurodegeneration that are seen in both flies and humans.
Thank you, Phil.
Scary prospect.
Yeah, I'm not sure what I've said tonight that isn't scary, sadly.
William, I notice you've got another question there, but we might pass over the worm compost question for the moment, and because we've got a couple more there. Kathleen, are you there, and would you care to unmute yourself and ask your question?
Yes, thank you. I've been using in recent times a repellent, I'm not sure if that's the right word for it, with garlic and chili in a base of vegetable oil and dishwashing detergent. What are your thoughts on that? It's diluted quite considerably to spray it.
I imagine it's safe. So I'd be quite happy to put that on myself.
Yeah.
But one of the things that I guess is a take-home message that I've not emphasized here is that there are many products that can be called organic that have not been tested to the same degree as other chemicals and could be, even more detrimental in the environment. Mm. So to take a chemical that's actually only been seen in nature once at a rum distillery in the West Indies and think that you can produce it in massive amounts and then use it all around the world is just crazy. Now, I'm not saying that there's not been research done on it. There has been research, but it's not been subjected to anywhere near the level of scrutiny that imidacloprid has been subjected to. And while imidacloprid is the demonized insecticide in the world, we don't know that it's any worse than other insecticides.
Yeah, we haven't investigated it.
Yeah, so that's why I think more of the work of the type that Felipe did needs to be done, because you can obviously, if you kill an insect with an insecticide, that's a huge issue. But if you're slowly draining the life out of a species by exposure to chemicals, then you end up with the same result.
Thanks, Phil. Musa, I wonder if you'd like to unmute yourself and ask your question.
Hello, thank you for a very, very useful lecture. I really enjoyed that. We are in the image of God. We are made in the image of God, but Drosophila is not in the image of God. But we are theologists, and we are thinking that we are the best. What's our duty of care about this climate change, about this all the things in this world is happening as theologians? How can we explain this to other Christians, that the importance of Drosophila is not it shouldn't be underestimated, or something like that? How can we do that?
Well, let me tell I guess there's two arguments I'd use, and one is biblical and the other is just biological. I think most people can understand the ideas of food chains and ecosystems. And I think we need to get away from the biblical language of species or kind and realize that there is a sophistication built into creation. And it is the interdependence that I talked about is really important. And if we as humans begin to mess with that, then ecosystems collapse. So that's the first point. The second point comes from the Bible itself, and there's two images that are really important. And one of them you've referred to, we're made in the image of God, it says in Genesis 1. And then the word used is that we are given dominion over creation. Now, that word is translated in various ways, but what I see is a delegated authority to treat creation the way God would, to love creation the way the Creator did and does. The other image I think is quite beautiful, and it's the image of Genesis 2, and you see God in this playfulness of being in the garden with Adam, and Adam naming the plants and animals, and you see the humans there as a caretaker of the garden. And I think that image is particularly useful too. So one delegated responsibility to behave as God himself would, and the other to be caretakers, loving it as God does.
Thank you, Phil.
Thank you for that.
Margot, your turn. Are you there, Margot? I'm looking for you and don't see you on my screen. Well, why don't we ask Margot's question anyway? Margot says some of our orchid species depend on various insects for pollination. Are insects in the bush declining too?
Oh, yes. So when I gave you an estimate of 10% population decline across terrestrial land insects over periods of decades, there's been a lot of research done in all sorts of environments in all parts of the world. Now, as time goes by, I think we'll get refined estimates as more research is done. But yes, We're not just talking about insects that live in close proximity to humans. And we're not just talking about climate change as a factor, but those other environmental disruptions that I talked about. One thing I didn't mention about imidacloprid that's just worthwhile bringing up in the context of this discussion, one of the reasons why imidacloprid is the top-selling insecticide in the world, and it's about 25% of the market, it and related compounds, is that imidacloprid can be used as a seed treatment. And so farmers can plant seeds that are coated in imidacloprid. When the seed germinates, imidacloprid is then translocated through the other parts of the developing plant. And this is where the impact on pollinators comes, because imidacloprid is translocated to the floral parts where the pollinators spend their time.
Okay. I think I'm audible now. Yes,
You
because
are.
I was wondering how the insecticides moved into the forest, but that would be one reason.
Yeah. Other ways, though, are basically if you're spraying, for example, then clearly it penetrates the soil, gets into water, and then it can move and be dispersed.
Thank you.
And that will be true of other sprayed insecticides.
Thanks, Phil. We have 2 votes now for worm compost. So we're going to go back to William and ask him about his question about worm compost. William, I can't see you, but if you're still there, you are. Yep. Why don't you ask your question about worm compost now?
Well, over time, why don't people just turn to using worm compost to fertilize their fields?
Thank you for that question, William. How old are you?
8.
8. I used to give talks in primary schools every year, and they were the best places ever to give a talk, because students in primary schools always ask fantastic questions. They're very imaginative, and they ask questions that adults don't ask. So yours is an example of a really good question. So the use of compost to produce good soil is an excellent thing to do. And, the use of chemical fertilizers, as you're picking up on, has been one of the factors that's doing a lot of damage in the environment. So that's a really good thing to do. And it's fun too, because you can have worms in your compost, and it's fun to watch them. Do you have compost?
Not at home.
No.
But we did try and we kind of forgot.
Right. Well, you've taught us all something tonight, so thanks for that question, William.
Good on you, William. Thanks for that. We have another question from Graham McLean.
Thanks, Chris and Phil. Look, the obvious layman's question, Phil, I think you're doing wonderfully important work as an expert scientist and operating from a terrific theological perspective. You've been delivering some pretty bad news, and the obvious layman's question is there any good news you can suggest? If I've understood, you've been posing the problem that we need the insecticides, but they're devastating, and the so-called good ones may be just as devastating as the so-called bad ones. Have you any thoughts, however preliminary? I know that as a scientist, you won't want to say something beyond what the science has proven, but however preliminary about how this problem is to be solved.
Okay. So in the longer term, we have to phase out insecticides, and we need to phase them out as quickly as we can. But as I've pointed out, we need an alternative. when I was a PhD student back at Monash, there was a very young honours student who turned out to be the best student I ever saw, and he's now probably Australia's leading biologist. His name is Ari Hoffman. And after his PhD, Ari went to California to study what was a really unusual phenomenon. There was a fly species, another Drosophila species, where there was this phenomenon of females becoming infertile, and it was spreading through the population. Now that's anti-Darwinian in a sense, because something that affects fertility should actually drop out of the population, not spread through it. He ultimately showed that what was causing this in these populations was not genetic, it was a bacterium that lived inside insects that could be transmitted. And it was called Wolbachia. Now, Ari, many years on, has done a lot of research on Wolbachia, and it can be used to control insects in a couple of ways. One of which is the way pretty similar to what he originally discovered. If you can get this Wolbachia into a species, and you can do that by actually injecting it, and then spreading it through a population, then you may well be able to bring those populations under control. Now, Ari and others have found that Wolbachia can have other effects. So there can be male sterility and a whole range of other interesting things that could be useful for insect control. But somewhat remarkably, the presence of Wolbachia can reduce the capacity of insects to replicate viruses. And so Wolbachia are being used now in many places around the world to stop the transmission or limit the transmission of diseases like dengue fever, Zika, chikungunya. And so this is really an extraordinary finding. And so I think that we need to consider things like that because there is a level of safety. Wolbachia does not really transmit from species to species. You need to get it, inject it, and then it will spread through that species if you release those insects. So it's a fairly natural solution, and it's not going to have any impact on the crops that we're trying to protect other than protecting them.
Fascinating. Thank you very much indeed.
One from Christopher Goddard. Christopher, are you still here?
Yeah, I'm still here.
Okay, go ahead.
Yeah, so it's a moderately open-ended question. I remember reading something somewhere that there's a fairly counterintuitive thing that you can do in terms of fisheries management, where you designate an area not to be fished, and then there's like a spillover effect where you can basically harvest off the top of that. That was used to great effect in various fishing communities, I think in Mexico fairly recently. So I guess my question is, obviously the amount of arable land, you quote that as decreasing every year globally. So we do need as much arable land as possible. But I guess, so to go back to the counterintuitive thing, could it actually be helpful to try to, I guess, to designate more spaces as wild spaces or managed wild spaces?
That's clearly valuable. To create refuge, basically. Very important. While I've talked about insects, and insects are incredibly important, as I've shown, in terms of being at the bottom of the pyramid, because the pyramid could easily collapse if you pull enough of them out. But it's true that other organisms need refuges too, and habitat destruction can be a huge problem. So looking at another area of genetics, if you imagine, an area that's, let's say it's got the shape of a rectangle, and organisms can move freely through that environment, then you can have quite a large population that may be sustained. If you then destroy the land in the middle of that rectangle, so that you've now got two smaller populations that are not connected, there can be real problems. There's a genetic problem that we often talk about, which is inbreeding. And I think we all have a sense that inbreeding can be problematical in humans. It's also problematical in animals. So if population size drops, then you have a big problem. So clearly maintaining refuge, not creating areas where you've got these little remnant populations that are likely to go extinct is very, very important. And in terms of the environment, something else that my friend Ari Hoffman did many years ago is just looking at the value in farms, with doing things like leaving some native grassland around the perimeter of the property. It increases insect biodiversity and turns out to have measurable impact on the productivity of the land that's bounded by this native vegetation. So there's lots of things that we can do, and reforestation is very important, and we recently had a fantastic ISCAST talk about that. I think something important that I've said tonight is that we can't just focus on climate change, although we have to focus on climate change. These other factors, use of chemicals, deforestation, etc., we need to be environmentalists and think about the environment in its totality.
Thank you. Good answer. Thank you.
Thanks, Phil. Mosh Jasper, are you there? I can't.
Hiding out.
Go ahead.
Hi, Phil.
We know each other? And
yes.
You've written a brilliant comment there. So do you want to give voice to that?
I was considering kind of speaking to the parts of it that might be, I guess to translate it a little, when you were talking about the organic, the two insecticides earlier, obviously one thing driving the selection of anything organic is this idea that somehow, with no respect to analyzing it, being organic makes something better for the world. And I guess one way I see that happening is that people often see there's an ideal the organic is connected to nature and things that are synthetic are connected to humans. Humans and nature are completely different. Humans are always the problem rather than the solution, which they often are. And so being organic is better. And I guess I was just bringing that against this ideal, there's this idea of pristine nature without humans as the good thing we're trying to get to. And obviously that's increasingly challenging as we move, as human population expands and cities expand. And I guess I was just thinking about the arc of the Bible, which begins in a garden and ends in a city that is a garden. It's not a city that, is a metropolis with no plants, no anything else. And I guess my question is, well, that vision, if we're thinking of solutions to these problems, one solution could be we run away from our humanity altogether. We just want humans to not be here. Or we might just throw nature out and want to just say, well, they're all going to die. We're going to, just go in the tech route. How do we, I guess I see at least the germ of a vision in the view of the garden to garden city of a way of living with the world that is more, that hasn't thrown technology, which in this case might be something scientific or synthetic out, but is living in it in a way that's still about flourishing and gardening. So I guess my question, putting that whole idea forward, your thoughts on that, particularly in the context of your research or your vision of what maybe this kind of flourishing looks like when it comes to insecticides long term.
Some scientists would say that climate change and other environmental degradation is not a problem unless you're worried about humans surviving. And the sense is that if humans go extinct, there'll still be a lot of species and evolution will continue and those species will adapt or not. So that's one point. I like your vision of the garden at the beginning and the garden at the end. And I recently conducted a funeral for a really dear friend who was a very keen gardener, and we in fact conducted the funeral because of COVID-19 in her garden. And I spoke about this image of her walking in a garden with God, because I actually, the Bible, some people have a vacuum cleaner view of what happens next in the world, and that is God comes and sucks people off the planet, and the planet, whoever's left, destroys the planet, and that's not a problem. But there's another vision in the New Testament which talks about a new heaven and a new earth. And there is a lot of talk about God, renewing the planet. And I think you can argue that we should be doing our best to preserve and be part of that movement for renewal. So I find that theology of thinking of heaven as a garden is really positive. In reality, none of us know exactly what heaven's like, but I find that really positive. And it seems to me that if this earth was the best that God could create initially, I'm not sure that there's a plan B that's better. So that's just my private thoughts, but I thought what you wrote there and what you've said is wonderful.
Thanks for taking the time. Appreciate it.
And a final, slightly easier question from Nicola there. Phil, could you please spell the substance that you were talking about in answer to Graham?
Which substance was that?
You were talking about dengue fever. It was the natural.
Oh, okay. It's not a substance. It's
with a W.
It's a bacterium. And
bacterium. So what is it?
And Moshi that I've been talking to knows a lot more about it than me, so I hope I haven't embarrassed myself. But he spelt it there. It's called Wolbachia.
Oh, okay. Thank you.
I'll give you a little bit more detail. So Wolbachia is one of a number of bacteria that are able to live inside the cells of insects. And so they are said to be endosymbionts. So they can live inside cells and they can have profound effects. And what Ari has found in his work is that depending on which insect you would inject the Wolbachia into, it can have different impacts. So sometimes you see these impacts on fertility, sometimes you see other impacts. And so in research, what they do is test different strains of Wolbachia and look at what their biological impact is and see whether it might be useful.
That's fascinating.
But I think it is the most natural solution.
And can it be used en masse?
Oh, yeah. So, clearly there are parts of the world where they are doing mass releases of Wolbachia infected mosquitoes. And so I think it's very exciting, but so far it's been largely used on mosquitoes, and I think there are many other uses, and we need to be investigating those.
Thank you.
With sheep blowfly, which is a major pest in Australia, because it's a parasite of animals, we can think about other strategies like vaccines.
I think it would work on sorry.
So my lab does work on sheep blowfly, and they parasitize sheep. And so you can think about the idea of giving sheep a vaccine that would allow them to create a hostile environment for the development of the blowfly.
Could you work on sandflies next?
On what flies?
Sandflies.
I'm having trouble getting research funding, so I'm not sure I can work on anything next.
Thanks, Phil. I'd love to tell you about my 3 months buckarooing in northern New South Wales and sheep blowflies, but it's a pretty not very pleasant.
It's an ugly story. I mean,
it really is, isn't it? Yeah.
I went to an international congress we call them sheep blowflies, but they're not sheep specific. And I went to an international congress of entomology, and there were talks about fly-blown humans.
Yeah,
Yes.
Okay, let's move on and let's finish with our young star of the night. William, would you mind unmuting yourself again and asking your last question? Because I think that's a great question.
Is there anything children can do to help?
Well, children can do something to help because you have tonight. You know, you've suggested a practical solution. And as you grow up, William, you will have more influence in the world around you, and you need to continue asking good questions and suggesting good ideas. When I go to a primary school and speak, I get more questions in a half-hour talk to primary school students than I've had in my whole career standing in a lecture theatre at Melbourne University. People stop asking questions. So, William, don't stop asking questions. Don't stop sharing your ideas.
Thank you, Phil. We're going to finish now. I'm going to hand back to Sarah.
All right. Thank you so much, Phil. It's been a fantastic talk. I've really enjoyed it, as I can see many people have. Just before we finish tonight, I'm going to get Dave Hooker to close in prayer. So, Dave.
Yep, sure, Sarah. I'm here.
Great. Thank you.
Thank you so much, Phil, and thank you so much to the ISCAST team for making this happen tonight. Let's just bow before the Lord in prayer to close. Father, we're very conscious of lessons that we need to learn as stewards of this world, and we're conscious tonight of the fact that we need to be far less anthropocentric in our mindset towards this earth and far more respectful and caring of all of your creation. Lord, we recognize that insects are part of your creation, part of the ecosystems, Lord, that you have also created, part of that interdependence and of which we are also a part. So will you change our attitudes, change our thoughts, change our perspective on how to care for your creation, change our sense of urgency, Lord, to make it much more urgent than it already is. And lead us also down the road, Father, to finding solutions, Lord, to this potential disaster, Father. We ask for your wisdom and your guidance in finding solutions that will help and not harm your creation. We thank you again for tonight. In Jesus' name, amen.